Okay, I've chosen a nice bright yellow background for obvious reasons. So we're going to talk about Fusarium wilt today, and some of the work that we've been doing over quite a few years in our lab over in the Heinz building. First of all, Fusarium wilt sometimes on banana comes in the news. There's always disaster stories, but it was first described actually in Australia back in the late 19th century, and but it's the other name you get for it is Panama wilt, and that's taken after the fact that it then appeared in Central America, in Panama obviously, and in areas round about there, and started encroaching on the banana plantations at that time, which that was when banana plantations and banana took off as a fruit of trade, because it coincide with the steam boats, etc., that were able to take the bananas quickly to North America and sell them to the markets there. So, and then you get some rather grubby stories happening because the people that owned the boats also started to invest in the land, and as Fusarium wilt took over, the land became contaminated because the fungus that causes it, it's a soil-borne pathogen. As the land became contaminated, these rather companies that were becoming bigger and bigger started a bit of a land grab. They would buy new land and displace the subsistence farmers, etc. And so a lot of the stories you talk about, banana republics, all that, and some nasty politics that went on with it, and coups and various things like that, all stems back to this fungal pathogen, Fusarium wilt. So it's got a lot to answer for, and if anyone's interested in reading more about it, there's actually a very good book that came out. It's called Banana, the Fate of the Fruit That Changed the World, because it has had a big impact, and the Fusarium has had a big impact on banana and politics. But anyway, let's move on. So at that time, Fusarium wilt was causing a problem in the start of the 20th century on all these new banana plantations, and particularly in Central America, but it really travelled, it'd been spread around the world, because if they knew then what we know now, they hopefully wouldn't have been moving the bits of banana plants or corms and suckers contaminated with the Fusarium around the place. Now, your grandparents may know, some of you, I don't know if there's anyone in the audience present, but would have, the cultivar that you would have bought in the supermarket, well, it wouldn't have been the supermarket, in the veggie shop, fruit and veggie shop in the early 20th century would have been Gros Michel. And people claim that it was actually a sweeter banana, more pleasant banana. You rarely see Gros Michel. In fact, I had to just grab a picture off the internet for that. And that was because it was susceptible to this first wave of Fusarium wilt caused by Fusarium oxysporum. And because banana is it's just a plant pathologist's dream. It's a monoculture, it's a single genotype, it's perennial crop, it's everything, and here you've got a soil-borne pathogen that gets into the soil and doesn't shift, it's there forever. And so you couldn't dream of a better scenario for a plant pathogen. And so Gros Michel became defunct. You could no longer grow it because so much of the land was contaminated. And so it was replaced. So we went through a period, and you may know the song, I'm not going to attempt to sing it, We Have No Bananas Today. I like to think that relates to that time, and it certainly coincides with that time, where bananas became quite rare to buy, to purchase, because certainly in the cultivated bananas, because of this problem with Fusarium wilt. So then along came Cavendish. Now, Cavendish wasn't a wonderfully bred cultivar that had been bred for resistance to Fusarium wilt. It was just coincidental. It was just a natural accession that had been taken and developed just somatically. There was no breeding involved. It originated, some stories say it came from Vietnam, other stories say elsewhere, but it'd been collected back in the 19th century by a chap called Williams, who was one of these early botanists that went out during the 19th century, was funded by by Lord Cavendish from England, and that's consequently he named the cultivar after his benefactor. And it was grown up, I think, in some glass house, maybe in the Cavendish estate or whatever, but it was also used in subsistence agriculture in Asia at that time. And it was discovered it was resistant to Fusarium wilt. And so as a consequence, 90% of the bananas that are grown, even more than 90% of the cultivated banana of trade now is Cavendish. In Australia, we have ladyfinger, and ladyfinger is still susceptible to that first wave of Fusarium wilt, but Cavendish isn't. So let's just look at banana cultivation worldwide, and 90%, I don't know the accuracy of the figures, but roughly 90% of banana production worldwide is for subsistence farming. It's not for trade. And in countries like India and Uganda, it's a very important crop. Now, roughly, I think it's 45% is cooking banana, but that's cooking banana and plantains are sort of categorized together, and the other 55% is dessert bananas, the sweet bananas that we're more used to. So only 10% of banana production is actually, or sometimes some figures say 15%, is for export or for commercial production. So it's grown in these vast, huge plantations, particularly like some that you'd see in the Philippines or in Central America and Ecuador and places like that. There are many disease problems because it's, as I said, it's such a beautiful crop for a plant pathologist, and a lot of aerial spraying goes on, not for Fusarium, but for other diseases such as the ones that cause black sigatoga. And if you live in some of these countries, you're constantly seeing the planes go over, dispersing the fungicides, etc. And they have wonderful problems with fungicide resistance as a consequence. So some of the disease issues, I'm just mentioning some of them, bunchy top virus, banana streak virus, Andrew talked about those a couple of weeks ago. Black sigatoga and yellow sigatoga. Black sigatoga is caused by a species, a fungal species known as Microsphaerella fijiensis. Its close relative, Microsphaerella musicola, causes yellow sigatoga, and we have that one in Australia. We don't currently have black. Oops, sorry, wrong way. I'm trying to find the pointer. Right. We don't currently have black sigatoga, but it is present in the Torres Straits, and we're always looking out for it, and a few incursions have happened. Freckle, this is one's caused by another fungus, Gignardia, and this is an image I just grabbed off the web. I hope it's a correct image because I haven't seen it, but it's just become an issue in the Northern Territory, and there's a big eradication program going on, which is going to have impact on trial sites, etc., in the Northern Territory. But this is also having a huge impact on the few remaining growers there, trying to stop its spread. Xanthomonas wilt we don't have, and that's a big issue in Africa at the moment. And it's a bacterium that causes this horrible oozy mass within the banana plant, and the banana will collapse, and we've got nematode problems. But Fusarium wilt is obviously my favourite. And Fusarium wilt's caused by the fungus Fusarium oxysporum. Now, you've probably come across Fusarium oxysporum before. Kamal talked about it last week, and particularly with in Arabidopsis, in that wonderful crop plant. And it's very convenient to work in Arabidopsis as opposed to banana for obvious reasons. But Fusarium oxysporum has a wide host range, but in host-specific strains. And we talk about term forma specialis referring to these host-specific strains. And it's assumed that something like Fusarium oxysporum forma specialis vasinfectum is the one that attacks cotton. Cubense is the one that attacks banana. And conglutens is the one that attacks Arabidopsis and also other members within the Brassicaceae. So to a certain extent, it's fairly tight host range, although I think it's always worth exploring that, because people tend to make the assumption if you collect it off a banana, it's a cubense one. If you've collected off cotton, it's a vasinfectum. And I think perhaps sometimes we need to test that. in addition to that, you also get non-pathogenic strains that are present in sometimes as endophytes in the roots of plants, but without causing apparently any harm. And so you can find them, and in some cases there's been a lot of work on a particular strain known as FO47, in particular against in tomato fields as a protectant against Fusarium wilt. So it's the same species, it's still Fusarium oxysporum, but it seems to increase the defense response, or it's even in some cases competitive with the pathogenic strains. So it's really a bit of a, you don't know if you're dealing with a pathogen or an endophyte or a biocontrol agent, so it can get a bit confusing. But just to add the fact that I couldn't get the picture actually small enough to put this to scale. So this is the Arabidopsis plant. It's cotton. We're dealing with this, and it's pretty big. It's got a fairly slow life cycle. It's a sterile crop. It's not exactly the easiest thing to work with. So there has been some infection studies done. We've done some in the past, but there's been some done overseas, etc. And what it's assumed that it's the same cycle that happens with most of the Fusarium oxysporums, that there's spores. It has 3 different types of spores. It can have chlamydospores, which are resting spores, and they tend to form when the mycelia runs out of nutrients, and the mycelia just starts to break up and form these these melanized cells, basically, that turn into spores. They also produce macro and microconidia, the classic banana-shaped fusiform macroconidia, and also the little microconidia, these smaller versions. Now, some of the earlier studies assumed that the microconidia were the ones that moved up through the xylem, because this is a vascular wilt. So once it gets in through the roots, it gets into the tissue, gets into the vascular system, it passes up through the vascular tissue. Whether it's the microconidia or whether it's mycelia, certainly when we've looked at cotton, Fusarium on cotton, we saw the mycelia growing quite happily up through the xylem vessels. But either way, these things are all present, and they can all lead to infection. When the initial infection occurs, the mycelia tends to accumulate on the surface. Now, this is just a GFP isolate. An isolate's been transformed with GFP just to show it. It's not the best picture, sorry. But the mycelia tends to accumulate on the root, and it's at that point, it's kind of like a mycorrhizae, and then sometimes we wonder if it's mimicking mycorrhizae in the way it tries to get into the root, and then it penetrates. Now, initially, the Fusarium is biotrophic, so it's a hemibiotroph, and it's not actually causing too much damage, and it's presumably gaining its nutrients in the apoplast, and then it then turns nasty on the plant and turns into a necrotrophic phase and spreads up through the xylem, leading to this sort of discoloration. As with any vascular wilt, it's the response of the plant that you see these internal symptoms by, because you're getting the plant is trying to stop the invader, so it clogs up its own xylem vessels using tyloses and also producing phenolics, etc. So you get this classic discoloration of the internal tissue, of the vascular tissue, and you've got the wilt. And as a consequence of that, the plant basically starts to shut down, and if you get that much xylem, whether it's consequence of the invasion of the xylem vessels or the plant's response to it, you get wilt, and the leaves start discoloring and the plant collapses, and you get no product, you get no fruit from that. So it's quite devastating, and in addition to that, that's not all. When the plant breaks down, the spores are released. The chlamydospores are thought to last at least 20-30 years. Whether that's the persistence in the soil or the fact that the fungus is still actually able to live as a saprophyte on the roots, decaying roots, or on roots of other plants, is maybe another reason that it's able for that long-term survival. And it's interesting to know that recently they started replanting banana up on the Atherton tableland, having been no banana there for 30-40 years, maybe even more than that, and it's come down with Fusarium wilt. So that it stays there, it's there forever. So it's a very serious pathogen. So the race terminology is a bit bizarre in banana, but the three races, as we talk about, that really matter. Race 1 is the one that came in that first wave that attacked the Gros Michel, particularly in Central America in the mid-20th century. It can attack both ladyfinger and Gros Michel, but Cavendish is resistant. Race 2 would attack Bluggo, which is more of a sort of cooking type banana. Race 3 apparently attacks Heliconia and has nothing to do with bananas, so it's a bit of a misname. And then we have the race 4 isolates, which will attack Cavendish. Now, we've had race 4 here in southeast Queensland since, I think, the early 90s, even maybe before, I'm not sure. And it will apparently attack, it's called subtropical race 4, where it attacks Cavendish bananas when they're in subtropical conditions, because bananas are tropical plants. So when we try growing it down here, it gets a little bit stressed over the winter time. You know that from the fruit. It loses that blush. It's a bit sort of has a matte color to it. And so the theory is that in the subtropics it's stressed, and so that's why it's susceptible to that. But having said that, nobody's actually ever taken the subtropical race 4 isolates and tested it in the tropics. So I'm not convinced that it wouldn't attack it, but because of quarantine, there's a lot of experiments that we can't do. But subtropical race 4 certainly is not as aggressive as what we now see tropical race 4, which will attack Cavendish in tropical regions, and it's a particularly aggressive form of the pathogen. It's become a huge issue in Southeast Asia. It's in the Philippines now, and more recently it's been picked up in Mozambique, which is a bit of a worry because it's now into Africa. It's in plantations there. It's not in subsistence agriculture as yet, but it's in a Cavendish plantation there, and it's been noted in Jordan, but I don't think they're so worried there because its chances of spreading in there is not such an issue, but certainly in Mozambique it is a big concern. So the distribution, Fusarium wilt, there's no chemical control. I mean, there's some attempts, but basically it's in the soil. You just can't get rid of it. There's been attempts where it's come in initially, digging up the plants and burning a big pit around them, but it doesn't seem to stop its spread. And there may even be, as well as being spores that be dispersed in the water around the plant, etc. There's some indications that maybe beetles can transmit it, even just short distances. I think some people have even blamed the geese in Northern Territory for spreading it around. I'm not sure. Usually human beings are the best vector, and moving plant material or moving equipment and stuff. So basically we have to rely on cultural practices for control and quarantine, and we do that pretty well in Australia. And this tropical race 4 turned up in Northern Territory in the mid-1990s, and it's been a concern ever since. There is a few farmers that are managing to still produce bananas there, but there's always that worry that the spores are good, that some soil's going to be transferred, down the highway, into northern Queensland, where the vast majority of our banana plantations are. So just to summarise, race 1's pretty widespread, both here within Australia and worldwide. It's not such a concern really in Australia anymore because we're mostly Cavendish. The ladyfinger growers, though, in northern New South Wales still managed to use a few fields that aren't contaminated, but that just that's probably the reason why you have to pay so much for your ladyfingers. Subtropical race 4 was really only ever an issue here in southeast Queensland and northern New South Wales. It's been in South Africa and also reported in Taiwan, but most of these are in subtropical areas. But it's tropical race 4 that has got everybody worried. It's in Northern Territory. As I said, we don't want it into North Queensland, where most of the production is. It's in much of Southeast Asia now. It's in Mozambique and Jordan. Central America are absolutely, and South America are absolutely terrified of it turning up there, because so much, particularly in the commercial plantations. So when you go up and down the highway, you see these quarantine signs. Much of it's for bunchy top as well as for Fusarium. But quarantine is, we're very fortunate in Australia that we can put these internal quarantine measures in, and they are reasonably adhered to. So we can't control with fungicides. We're trying to control it culturally. The obvious thing is to get a new resistant cultivar to this tropical race 4. But that's a bit of a problem. Cultivated banana is a sterile polyploid with a parthenocarpic fruit. It's a total freak of nature. It shouldn't be there. It's only there because of us, because we cultivate it clonally. We take bits and suckers and we transfer them. Now we can use tissue culture, so it's a little bit cleaner. But basically, the cultivated banana wouldn't survive without our intervention, and it's only been presumably selected from the wild because it doesn't have any seed in it. Now, I haven't got a picture of any, I didn't get a close-up picture of seed, but there's some here, the dried up ones that were in my office if you want to have a look at them later. But if you were going to crunch into a banana with seed in it, it wouldn't be very pleasant. It'd be a bit like pomegranates, I guess. But so that's not only a problem when you're trying to breed it, but also when you're trying to breed one, your end product, you want it to be sterile. So you don't want to, you want to get rid of the seed productions phase of it. The bananas we eat are mostly from Musa acuminata, the A genome, but also sometimes the B genome, Balbisiana. And it's actually thought the Balbisiana has a lot more resistance in it, and it would be nice to introduce a little bit more of it, but then you have added problems when you start doing crosses. So ladyfinger has a bit of the B genome in it. Cavendish is apparently mostly just the A genome. And so obviously they're polyploids, which makes them a little bit bigger anyway, but they're sterile, they've got uneven chromosomes in them. And compared with the wild diploids that produce fruit, I think that's the next picture, yeah. The wild diploid tends to be produce smaller fruit, but lots of seed, which can be a bit of a problem. And isn't necessarily parthenocarpic. So parthenocarpy is when you're getting fruit without the seed again, which is of no advantage to the plant. So it's extremely difficult to breed for resistance and to get this end product of parthenocarpy and sterility. And on top of that, you've got long generation time, a rather large plant, and you may have problems with interploidy crosses. And in addition to that, the people that have done the banana genome discovered that there's quite a large proportion of the genome is made up of transposons, just to make life a little bit more confusing as well. So there have been some breeding efforts with using improved diploids crossed onto triploids and then scouring through lots and lots and lots of banana fruit to get the occasional seed that may recover and then take that through a screening program. Much of that type of breeding was done in Honduras, and I guess labour costs are a little bit less than they would be here in Australia, but even still they haven't only recently now we're seeing cultivars coming out of these breeding programs. Goldfinger was one such cultivar, and it did have some level of resistance to Fusarium wilt, to race 4, but it's not been commercially acceptable for reasons. They say the consumer doesn't want it, but maybe sometimes it's maybe the wholesalers don't want it because it has different ripening requirements, and so it makes it a little bit more complicated than just having to deal with Cavendish. So as yet, we have no commercial cultivar with complete resistance to race 4. So along comes Musa acuminata, subspecies malaccensis. Now, it's been long known that it does seem to have some resistance to Fusarium, and you can see they're a much skinnier plant than your average one because they're diploids. I'm sure it's nothing to do with us not tending our plants very well, but they're much skinnier plants, but they do seem to, now initially, this, we got seed from this line was a scientist, Ivan Budenhagen, he's emeritus at the University of California, Davis, was on a field trip with Susie Bentley and Natalie Moore, who used to be here within the CRC for tropical plant pathology. And Ivan discovered this wonderful, healthy, wild banana growing in a devastated plantation of tropical race 4 in Indonesia. And so, being an old school scientist, he grabbed the seeds and brought them back to Australia, which they was allowed to do at that point. And the seeds were embryo rescued by Sharon Hamill at the DPI in Nambour, and her and Mike Smith ended up putting it out into a plantation in southeast Queensland where subtropical race 4 was an issue. And they noticed, and that's when we got involved, that there seemed to be this lovely 3-to-1 segregation within this population. Even though it was a bit of a mixed parental population, you were seeing this segregation going on. So plants that were dying next to wonderful, vigorous, healthy plants. Now, you can't take too much from that because Fusarium's a real pain in the neck to do in field trials. You need to do lots of replications of individuals because you can get misses. You do that with any soil-borne pathogen, it can be an issue. But anyway, the fruits are small, they're seeded, and they're of no commercial value, but they have that potential resistance in them. Some of them, coincidentally, are also parthenocarpic, which makes it interesting too, so worthwhile following up. So we took, so there were signs of single gene resistance, so that's where we got excited about it. So from doing embryo rescue, those original 40 plants who had this apparent 3-to-1 segregation, at that time there wasn't that much funding around, so 3 resistants and 3 susceptibles were taken back into tissue culture and maintained. And this has been quite a while, and so we've just been ticking it over the years. And we had some plants originally at the UQ's farm at Redlands, and then when that closed down we moved them to the DPI Redlands farm. So we still have some plants there, and occasionally we'll go down and would attempt crosses between the resistant and susceptibles, or I've done self-pollinations. And so just to let you know, it's not that simple doing, I mean it's quite surprising when we look at banana how little has been done on basic genetics, because I guess the aim has been, particularly with the banana companies like United Fruits, was to get a product. They weren't interested in the genetics, they just wanted a more vigorous plant, a more resistant plant. But very little has actually been done on classic genetics, just good old Mendelian genetics, which I find fun. And if you take an inflorescence of banana, the flowers that first start up, these are the oldest ones that would have been up here, are the female flowers, and so they will receive the pollen, and then you get the bunch forming there. And as each bract opens up, it reveals another hand of flowers, and as you go down the bract, the pollen becomes, the male flowers become more predominant, though you can still see residual sort of stigma, etc. But so you have to sort of take the pollen from there and pollinate them. So we've done that over several seasons. In southeast Queensland it's best done in the summer months, although we can still get some seeds over the winter. Further north you'd be able to do it all year round, I'm sure. And so we've harvested seed from that, and we've embryo rescued and grown the plants up, and again these skinny little things here in the glass house. And then we challenged them initially with subtropical race 4. We can't work with tropical race 4 here. We don't want anything to do with tropical race 4 in Queensland. But so we would do pot trials, and with, again, as being a soil-borne pathogen, you need to do lots of replication, but that's one thing with banana, you can easily clone it. So we'd clone each line, and so have at least between 3 to 6 plants per accession, and then screen them with subtropical race 4. And that was a sort of classic sort of wilt symptoms. And in fact, it actually happens very quickly with these malaccensis. The malaccensis susceptible plants tend to be more susceptible than a Cavendish susceptible or a ladyfinger susceptible. So you'd see symptoms quite early on of wilting compared with the lovely healthy ones which were resistant. And then if you look at another classic symptom is this stem splitting. You see this in the larger plants, but again we see it in pot plants. And then the discoloration of the rhizome when you open it up. We did inoculations by infesting millet, autoclaved millet with the fungus and incorporating it in the soil. There have been various ways of doing it, but we've found that that's been the most efficient. And when we look at those 6 original, I've only got 4 of the parents here, 1 of the susceptibles and the 3 resistants, we get conveniently all the susceptibles were all produced progeny that was all susceptible. 1 of the resistant parents produced progeny that was all resistant, and 2 of them appeared to be heterozygous, producing progeny in roughly a 3-to-1 segregation. Now, you might say, didn't she do more plants than that? I wish we did. We did actually have more seed, but there was various tissue culture room failures which reduced the seed numbers, but also we've got to remember we've got to replicate these each accession and grow them in the glass house, and you can only inoculate the plants once they're about 16 weeks old, and then symptoms take 10 weeks on top of that. So it's not exactly as convenient as Arabidopsis, put it that way. So we got this result with subtropical race 4. So, I mean, we're interested in that, people in southeast Queensland, but the rest of the world is interested in tropical race 4. So we wanted to repeat that. So I say we all the time. This was mostly done by Sam Fraser-Smith, a PhD student with us. So he wanted to repeat this, and but with tropical race 4. And we have tropical race 4 in Darwin, and so through a lot of logistics we got plants sent up in tissue culture stage to Darwin, and with collaboration with the Northern Territory DPI, these plants were then grown up, put in a shade tunnel, because they didn't have sufficient glasshouses for that. We hoped we would dodge cyclones during this time, and then Sam went up and inoculated the plants using the same method as we had done here in Brisbane. And more or less we got the same result. Obviously it was a smaller sample because of the expense of taking stuff up there, but again we're roughly getting that 3-to-1 segregation. So the numbers are too small for a chi-squared test, so I can't confirm that. But certainly the same plants, the same individual plants, the same clones that were resistant to subtropical race 4 were resistant to tropical race 4. So we think we're dealing with the same gene to that, or at least it's closely linked, if not. So the next question was, what about race 1? And this is where it was quite interesting, because again we see segregation going on there against race 1. I'm not convinced, because we've got too small a sample and we need to repeat this, whether we're dealing with the same resistance as the tropical race 4 resistance, but it is important that we are seeing resistance and susceptibility within the race 1, because as I'll go on to say why race 1 is important. So we've got pretty good population there. So the next thing was obviously get hold of the gene. So if you're dealing with a clonally propagated crop that's so difficult to breed, well, in an ideal world you grab the gene and stick it into Cavendish or stick it into Gros Michel, if that ideal world would accept your bananas, that is. But regardless of that, we've got a good molecular marker for a breeding program. So what we're now doing in collaboration with Jackie Batley's group, which is still just here at UQ, but soon to move, we've been looking at this population and using a genome-wide association study correlating SNPs with resistance and susceptibility to try and locate that resistance gene. Now, conveniently, the banana genome has well now been sequenced, or not fully sequenced, but certainly been published, and so we can tap into that information to try and work out where our chromosomes are and what, etc. So looking at, we've identified SNPs in the various chromosomes in the banana genome, in the malaccensis lines that we're dealing with, comparing resistant with susceptible bulks. And then by looking at the genome, Jackie's group, they've been able to identify potential resistance genes that are based on the published ones of leucine-rich repeats or receptor-like kinase genes, and there's 302 apparently potential resistance genes within that genome. but that's assuming that our resistance gene is that type, but let's just assume that at the moment and not make life more difficult. And some of them are still in the unmapped regions, and that indicates that the genome isn't completely sequenced to perfection as yet. And what we've found is that by comparing the resistant and susceptibles, if we look at the blue lines, that's just comparing the heterozygous resistance, so it could be big R, little r, versus 2 little r's. You're seeing SNPs, so you're seeing differences along the chromosomes. But if you look at the little orange spot here, this is a sort of a hot spot which is looking at difference between homozygotes. We've got less proven homozygotes. We need to do more plant screening, but we can compare big R with little r's, basically. So we're looking for presence or absence of SNPs, of single nucleotide polymorphisms in the DNA for differences there. And we seem to have a hot spot here at the end of this particular chromosome. And that's where you expect resistance genes to be located. They're often telomeric, and certainly the ones that have been identified so far. But to tighten that up, we need a lot more. We need to screen a lot more plants. So we've increased the malaccensis plant. We have more than 400 plants available now that Liz, somewhere in the audience, has been busily tissue culturing in our tissue culture room and bulking them up so that we can have more plants to screen. But as I said, it's not that easy. We need to find a glass house that somebody will let us contaminate with our Fusarium, and it takes at least 26 weeks to go through the full process of this to test one plant, and then you've got to multiply them up, because with Fusarium you have to replicate it in case of misses. So it's a long procedure, but we still have plants in the field, so we're going to increase that. But the good news is that in collaboration with IITA in Tanzania, we've managed to get some Bill and Melinda Gates funding to increase this population and do the phenotyping. In East Africa, race 1 is a big issue in with the cooking bananas, and particularly in Tanzania and Uganda. So they're interested not just in the race 4 resistance, but also in race 1 resistance. So and for mapping in their breeding programs, so that we can get hold of nice fine molecular markers to incorporate into the breeding programs and to introduce the malaccensis population that we've been working with into the breeding program, it'll be quite exciting. And also, obviously, as a GM, there's potential there that if we can get hold of the gene and ideally transform it into Cavendish or Gros Michel, the commercial banana production would be certainly grateful for that too. So that's the ongoing things that we're doing with the plant. Now, I'm just going to have to speed up because I want to talk about the pathogen as well. So we're looking at that too. Now, Fusarium oxysporum cubense can be categorized by race, which I've talked about before. There's also volatiles. You used to be able to distinguish race 1 and race 4 by sniffing it, because there was different volatiles given off. Apparently that's not concrete, that one. People are mostly reluctant to do that. I'm sure that we would have, if they did some gas chromatography, they could work it out. But anyway, another way that Fusarium oxysporum, as you find with some other fungi, they're categorized by the VCGs, the vegetative compatibility groups, and also phylogenetic clades. Well, let's look at VCGs. VCGs are where you've got fungi. Now, these are asexual fungi, and when the nuclei can transfer from different individuals within the species to another individual within the species. Now, sometimes there's a barrier and that doesn't happen. There's no exchange, there's no intermingling of the mycelia, there's no exchange there. And that's thought that that barrier has possibly evolved to stop the spread of mycoviruses within a fungal population. So anyway, it's a convenient means for us to look at close relatives of these individuals. So we say they're within the same VCG group, where you get complementation in a chemical pathway such as the nitrate reductase pathway. So initially you have to create mutants within that pathway and then see if they'll come back together, and if it's in different steps in the pathway they'll complement each other and allows them to grow on a media that breaks down what's actually chlorate media, which allows them to grow on that. So it's not an easy process. It can take several weeks just to see if these two isolates are genetically related. So obviously we've moved on from that. We can use molecular biology also to see if they're genetically related. But VCGs, and Fusarium oxysporums have been identified based on their VCGs. And then people have gone on to use molecular tools to identify them. And whether it's ITS. ITS doesn't always work that well for Fusarium. It's thought because of the chromosomes, because of the transposons in it. They've used elongation factors, looked at sequence analysis. But all the time you find it doesn't necessarily follow a nice tight phylogeny. And so we always talk about them being polyphyletic, that its ability to attack banana has evolved more than once, because you find race 1 isolates of FOC here intermingled with, there's lycopersici which attacks tomato, there's chrysanthemum ones, there are different lots of things. And here we've got race 1 isolate down here along with race 4 ones. So it's all intermingled, all interspersed. And so the story didn't tie up very well. And so along came a chap called Rep and his group in Amsterdam, and they started looking at Fusarium oxysporum lycopersici, which is the one that attacks tomato. And I know Kamal made mention of this last week. And it turns out that these 6 genes, which they had identified by reverse genetics going into the vascular tissue of these infected tomato plants and finding what proteins were there and working back to the genes, and that's why they called them secreted and Xylem genes, turned out to be actually the effector genes, the avirulence genes involved in the tomato-fusarium interaction. So everyone got quite excited about that. So they identified 6.1, 3, and I think it's 6.4 I haven't mentioned there, initially were involved in those avirulence genes. And it turns out that many of these 6 genes are associated with a dispensable chromosome within the Fusarium oxysporum genome. So the obvious thing was to see, well, are they in the cubense? And so a student that we had in Northern Territory worked alongside Sam, and we looked to see if any of the 6 genes were present in the Fusarium oxysporum cubense isolates. And we found, 6.1, which now all these 6 genes, secreted and Xylem genes, it's not part of a gene family. Their only commonality is they're secreted in the xylem, so it's not one biochemical family of proteins that they produce. So but 6.1 was found to be present in all the FOCs, but interestingly enough, it wasn't in, although we're only looking at 3 of them, 3 of the non-pathogenic Fusarium oxysporums that we had obtained from a banana field. So these were Fusarium oxysporums that are present but without causing disease. So it was interesting enough to know that they didn't have these pathogenicity genes. And then we looked at some of the others. Again, Rachel and Sam looked at some of these others, and they found that 6.7 and 6.8 was able to discriminate between race 1 and race 4 isolates. So that if we've got, I think it's, the green is tropical race 4, and the purple, which I can barely see here, is actually subtropical race 4. So you see these bands with PCR was associated with them. So that was using PCR. Being an old cynic, I said we better check with Southerns, and the same thing happened. That we still got the same sort of banding pattern when we did Southern blots to identify the presence or absence of these genes. So that we could see an association, we could see a difference between race 1 and race 4, which makes it interesting. So then we upped it, and again, linking in with Jackie's group, we started using next-gen sequencing. And the one advantage, we have access to a very wide, large collection of Fusarium oxysporums which is held at DPI. So Wayne O'Neill was very generous with his space and time, in that Sam would go over and extract DNA from these isolates and start screening them. So, and then Liz Czeslowski has did her honours and had started a PhD at the beginning of the year, has also become involved in this project, and she's been looking at pathogenicity genes across a whole range of these isolates. So just by screening the database, seeing what's implicated in pathogenicity, but putting in one of the 6 genes there too. So, and these are the sort of isolates that we've screened, representing numerous numbers, representing a whole lot of different VCG groups, including non-pathogenic isolates. And when we look at the different pathogenicity genes, we find representatives of them across all these isolates, including the non-path isolates. So there's still there's SNPs, there's slight variation in the sequence, but they're still present. But then we look at 6-8, and we find it's not in race 1 and 2, and it's not in the non-path, but there is a slight variation in sequence between races, subtropical race 4 and tropical race 4. And so that's useful because it's useful for a diagnostic. Some in the past, a lot of the diagnostics based on the ITS have been unreliable, and probably they've been unreliable because of what's going on with this fungus is that there's horizontal transfer going on with pathogenicity genes, because these 6 genes, and now this is the work of Ma et al., where they started looking at Fusarium, oxysporum, and particularly lycopersici, and they did a comparison, get rid of the stars for a minute, did a comparison between other Fusariums, and they found that there was a lot of conservation with many of the core genes, core chromosomes, but chromosome 3 and chromosome 6. If you're looking at comparison with other Fusarium species, there's a lot of variation there. But they also talk about this dispensable chromosome, chromosome 14, which seems to carry a lot of these secreted in Xylem genes. And if we look at the path genes that Liz looked at, they're dispersed across the core genome, but nothing in chromosome 3 or 6, or nothing in chromosome 14. But when she looks at 6, 8, we find it some occasional ones on a core genome, but they tend to find these are copies on the dispensable chromosome 14, and some on 3 and 6 as well as an individual one elsewhere. So there's sort of further indication that there's perhaps horizontal transfer has been going on. It's been transferring these pathogenicity islands or specific chromosomes from one individual to the other, and that might explain why we're getting this polyphyletic appearance of the isolates that will attack cubense, attack banana, so that they're not genetically related. It's just they've been exchanging pathogenicity genes, and that's what's allowed it to happen. Okay, I'll just flick through that. So what we've looked at so far is 6, well, we've actually looked at more, but I'll just talk about these ones at the moment. 6.1 and 6.2, and we can discriminate on the basis of absence adherence on race 2 of 6.2. 6.6 is in race 1 and tropical race 4, but not subtropical race 4. And 6.7 and 6.8, as we saw before, was present in these 2, but because there's a sequence variation, is enough to discriminate them. So it's involved, so there's a fairly strong association that's involved in pathogenicity. We haven't proven that yet, and hopefully Liz is going to do with knockouts and transgenics. We will try and manipulate some of these isolates to see if there's a gain of function or loss of function with transgenics. And so we'll continue to do. So we know that 6 genes can be used to distinguish, so it's a useful diagnostic. And also we want to look at the interaction, the actual action of these 6 genes to see how important they are in attacking the banana plants. Why is tropical race 4 so aggressive? So these would be reasons that we'd want to look at it. The other thing which we're interested in, and we will follow up, is that how stable are the forma specialis? Occasionally we come across this thing called the false Panama, where a banana plant suddenly comes down with a Fusarium wilt, but when you look at the isolate, the VCG, it doesn't seem to be the one that should normally attack that particular cultivar. So so that there may be something going on in the field where there's a interaction with a non-pathogenic one that might be carrying a pathogenicity factor. So there's a lot that needs to be dissected. And also we're moving out banana, we're looking at other crops, and people will come across, we're interested in ginger as well, which also gets Fusarium wilt, and they'll come across a ginger field where previously there was watermelon, and the watermelon had Fusarium wilt, and then the farmer will say, oh, I've never seen such a problem in my gingers since I got the Fusarium wilt in my watermelon. So we're going to look at that, even though the watermelon isolates are supposedly quite distinct from the ginger isolates, to see if there's complementation or something is going on there. So I think, although we've been aware of Fusarium oxysporum causing problems in horticulture since the end of the 19th century, I think there's still a lot we can find out about it. Okay, so in summary, we've got single gene resistance to tropical race 4 of Fusarium. We'll follow it up with molecular markers for breeding and possible candidates for GM. And with the 6 genes associated with race specificity, we have a molecular diagnostic that can be used, all importantly, to see have you got tropical race 4 present or not, and possibly sort of look at it more interesting at the scientific perspective, see if there's a gene for gene going on, and to follow up from there. Okay, and if you notice, if anybody noticed the color change of the slide presentation, we've got to the point where the color of an overripe banana, and I'll finish there. Thanks. Oh, and the acknowledgements, the also important acknowledgements, very rude of me. Much of the malaccensis work was done with Sam, with various honours students and people in Jackie Batley's group, as well as Sharon and Mike from the DPI, and initially that seed from Ivan Budenhagen. And we're very grateful that it's coincided with the point that the Banana Genome Hub or Consortium has come up with the banana genome based on the malaccensis cultivar, but not the same as ours, which has been very useful. And the pathogenicity work has been done also with Sam and Elizabeth Czeslowski, Katerina did some, she should be in that group, embryo rescue and interactions with Julian's group as well as people in the Northern Territory, and Wayne over at DPI. And I haven't mentioned, and apologies, it's okay, Kevin's not here, I haven't mentioned our work with silicon, looking at if we can't have genetic resistance, if we can look at cultural control or certainly enhancement of plants to defend themselves against Fusarium, we've been looking at silicon applications, and also in the past we looked at biocontrol possibilities for trying to reduce the level of Fusarium. Okay, I'll stop now. Thank you.